Auzubillahiminash-shaytanir-rajeem. Bismillahir-rahmanir-rahim. Welcome to International Conference on Applied Zoology, which will held in the Khwaja Farid University of Engineering and Technology Sciences, Rahimyar Khan, in the cooperation with the Applied Zoological Society of Pakistan. I am Asif memon, PhD scholar, Department of Zoology, University of Pakistan. I will deliver my oral presentation on the biodiversity and taxonomy of long-horned grasshoppers and short-horned grasshoppers from the district Dadu Sindh. These are the contents of my study: introduction, objectives, material methods, results, conclusion, references, publication, and acknowledgement. First of all, introduction to the biodiversity. The biodiversity may be defined as a biological variation and variability among the organisms. The biodiversity term was given by Walter G. Rosen in 1985. More formally, biodiversity is comprised of several levels, starting with genes, then individual species, then communities of the creature, and finally an entire ecosystem. Introduction to the taxonomy. Taxonomy is the knowledge about the classification, identification, and distribution of the various organisms to various groups. This term is derived from the Greek word taxis, means arrangement, and norms means law. The taxonomy is therefore methodology and principles of systematic biology and set up arrangements of the different kinds of organisms on the earth. Grasshoppers are the potential pest of all plants, trees, and most serious enemies of the agriculture. Moreover, grasshoppers attack not only the agriculture crops but also rangeland fields, and they cause considerable loss to our valuable crops. The short-horned grasshoppers are common in moist and swammy areas. These serious pests has been recorded from every part of country throughout the year. Some grasshoppers cause significant damage to the tree seedlings and agriculture crops. They are widely distributed and absent only in truly polar areas and some few small group of islands of the oceanic origin. They are hemimetabolous insects, means having incomplete metamorphosis. They are included in oligophagous and mixed feeder. They are functionally very important ground invertebrates in grassland ecosystem. Many species are green or straw colored, which help them to blend in their surroundings. They are herbivorous and include some of the most destructive agriculture pests known. A female short-horned grasshopper lays about 100 eggs in the soil. The eggs hatch after a rest period, and newly hatched nymphs, miniature replicas of adult, pass through a series of mold before they become adult. And the series of change is called metamorphosis, and I already told you about the metamorphosis occurs in these insects, which is incomplete metamorphosis. They are often the main invertebrate in grassland ecosystem for consumers, and they are also important food source for the many group of predators, for example birds and others. This is the general structure of short-horned grasshoppers, including head, thorax, abdomen, mesothorax, the antennae, the protosternum, mesosternum, pronotum, and all other these parts of the body. These are the objectives of my study: to collect the large number of short-horned, long-horned grasshoppers from the different selected localities of the district Dadu, to identify the collected material up to subfamilies, genera, and species level, and new record and new species. To prepare the taxonomic key for the subfamilies, genera, and species. To prepare photographs with the illustrations. The material methods. This is the map of district Dadu, and district Dadu includes the 4 Talka Meher, Talka Kensha, Talka Johi, and Talka Dadu. So this map showing the 4 talkas of the district Dadu. This is the methodology. The specimens were brought to Advanced Entomological Laboratory Department of Zoology, University of St. James, and zoological name lab of Government Ustad Bukhari Degree College, Dadu. The specimens were examined in the lab, samples pinned and examined their important morphological characteristics such as head including antennae, thorax, pronotum, prosternum, mesosternum and abdomen and other the main important parts of the body. This is the methodology. First of all, collect the specimens, then kill by the killing process and then pinning, then stretch the specimens on the stretching board, then preservation takes place and after the preservation the examination takes place. This is the list of equipments which I used during the experiments during my study. These are the list of chemicals which are used in the sample preservation: chloroform, naphthalene balls, alcohol and killing insect jar. This figure showing the study area during the sample collection, the results of my study. There are 247 specimens collected from different areas of district Dadu. The specimens were identified into family activity, the 7 subfamilies, 12 genera and 15 species. So this table showing the classification of these specimens into family, subfamily, genus, species. The family activity, these are the subfamilies: Oxini, Hemiacrididae, Podipodini, Menonopodini, Acridity, Gamosphonie. These are the families, these are the genus and these are the species which I collected during this table showing the collection of species from various localities of the district Dadu. This table showing the highest number of these species Oxia hyla hyla reported. The Oxia hyla hyla reported is the maximum number in the selected localities: village Fakka, village Qazi Araf, village Muhammad Ibrahim Pawar, village Ghulam Hussain Gadhi and village Bachalpur. The least number of the species which I collected from during the study, which is Enacrydium recipientum. This graph showing the collection of species from various localities of district Dadu. This table showing the incidence of the short-horned grasshoppers from various localities of district Dadu. The present studies carried in 2021 and 22. Percentage-wise data is presented as below. The highest percentage was recorded for the Oxia hyla hyla, which is 19.02, and the lowest percentage was recorded for the Enacrydium recipientum, which is 2.02%. This is the graph showing the percentage of species found. This is the conclusion. As a result of present study during the 2021, I have collected 247 specimens. These specimens belonging to family Acrididae, short-horned grasshoppers. During the present study, 15 species are described. The most abundant species is Oxia hyla hyla cervidi. Of the total number of all species, followed by Oxia phosphata, Herocleptus purpurata, Ilopus thalassenius, Ilopus thalassenius timulus, Locusta migrata, Spenicus cervici, Trilophidia annulata, Ecrata exculata, Gonesta rundetata, Enacrydium recipientum, Helithera olipedes, Eclotylus insabricus, Eclotylus fisteri and Melanopilus differentius. Oxia hyla hyla is considered as a major pest of rice and found in all selected localities of the district Dadu. So these are the key of the subfamilies of ability. More keys. These are the publications. The research paper published in the Journal of Animal Sciences, University of St. Jamshoro. This paper published in the Journal of Global Biosciences. Another publication. These are the certificates. More certificates. The certificate from the Zoological Congress. These are the abstracts. References. More references. Acknowledgement. I am thankful to Honorable Dean Faculty of Natural Sciences, Professor Dr. Wazir Baloch, and the respected chairperson and my supervisor, Professor Dr. Naid Baloch, and my respected co-supervisor, Dr. Rifat Sultana, and all the respected teachers and research fellows of the Department of Zoology, University of St. Jamshoro for their kind cooperation and guidance during my study. So I am thankful to all. Thanks a lot.